Hello friends, myself is Assistant Professor Rekha Jadhav, HOD of Zoology in VN Knight College, Nashik. Today we'll discuss about countercurrent exchange, is also known as a countercurrent multiplier theory, as a part of physiology of excretion. So friends, we know the basic structure of nephron. Nephron is a structural and functional unit of kidney. It shows different parts like first part is a Bowman's capsule, which is a cup-shaped part in which there is a dense network of blood capillaries called as a glomerulus. Next to the Bowman's capsule, there is a convoluted tubules called as a proximal tubules. Next to that, a U-shaped tube is there, which is called as a loop of Henle's. Next to that, distal tubule, and the last one is a collecting duct. Through the Bowman's capsule to the glomerulus, there is a filtration of blood, means urine is formed. When that filtrate runs through the PCT, loop of Henle's, and DCT, there is a reabsorption of water and salts through a specific way. So when water gets reabsorbed through the loop of Henle's, at that time it absorbs in a specific manner and it is called as a countercurrent exchange. It is also known as a countercurrent multiplier theory. Now here we can see the descending limb of Henle is permeable for the water and through that water get reabsorbed. But ascending limb is not permeable for the water and through that only salts get reabsorbed. Collecting duct is also permeable for the water and water get reabsorbed. But this reabsorption is carried out in a specific manner. See here, the outer U-shaped tube is a loop of Henle. First one is a descending limb of loop of Henle and here is a ascending limb of loop of Henle. And in between these two, there is a U-shaped tube which is a vasa recta, is nothing but a blood capillary. Now see, when filtrate or the urine flow from the loop of Henle to the descending limb, at that time water get reabsorbed and therefore the concentration of ions is get changed. And see, the concentration of filtrate is first of all 300 milliosmoles, then 600, then 900, and it reach up to the 1200 milliosmoles. It means because of water reabsorption, what happen? Concentration of salt get increases. As this filtrate flows from the ascending limb of loop of Henle's, there only salts, means sodium, potassium ions, they get reabsorbed, and because of that filtrate become hyposmotic. It means concentration of ions get decreases and its osmolarity from 1200 to it become 900, then 600, then 100 milliosmoles. Now at the same time, see the ions which are reabsorbed from the ascending limb of loop of Henle's, they get enter or get absorbed in a vasa recta, and due to that blood which is flowing through the vasa recta, first of all it is having concentration 300 milliosmoles, then it becomes 500 milliosmoles, 600, 900, and finally it becomes 1200 milliosmoles. Means due to the absorption of salts in a blood, its osmolarity get increases, its concentration get increases. When that blood flow from the another capillary or from the part which is nearby to the descending limb of loop of Henle's, water is reabsorbed from the descending limb of loop of Henle's, and due to that concentration of salts in that blood is get decreases because of water entry. So the concentration becomes 900, 600, 500, and finally 300 milliosmoles. It means osmolarity is maintained in the blood capillary as well as in the loop of Henle's, and it is called as a countercurrent exchange. Even same is known as a countercurrent multiplier theory. Thank you.